Hello, and welcome to this Nature Custom Media webcast titled Autoantibodies as Biomarkers for Cutaneous Melanoma. My name is Nikki Forrester, and I will be your moderator. Today's webcast is produced by CDI Labs. Autoantibodies are emerging as promising biomarkers for many cancers, including cutaneous melanoma, offering potential advantages in early detection, prognosis, and treatment monitoring. These antibodies target tumor-associated antigens and can be detected in the sera of melanoma patients, often before clinical symptoms appear. Today's webcast will highlight novel technologies that enable the detection of autoantibodies by screening human serum samples against more than 80% of the human proteome. The presence of autoantibodies may indicate immune recognition of melanoma cells and correlate with disease progression, response to therapy, and occurrence of treatment-related side effects. While autoantibody panels could enhance diagnostic accuracy and disease monitoring, challenges remain in standardizing these biomarkers due to variability in their data analysis pipeline, immune response variability, and specificity concerns. Future research integrating autoantibody profiles with other molecular and imaging biomarkers may improve melanoma detection and patient outcomes. We'll begin the webcast with a presentation from Dr. Pauline Zaenker of Edith Cowan University. We'll end the webcast with a Q&A. To ask a question, just type it in where it says Ask a Question and then press Submit at any point during the webcast. We'll do our best to answer all of your questions today. And now, over to Dr. Zaenker.
Thank you so much for your introduction and also in general, just for the opportunity to present on this important topic. Here are my disclosures for this presentation. Now, blood is an extraordinary medium that can be easily and repeatedly be collected for medical research. And the development of blood tests that are capable of detecting a diverse set of biomarkers has significantly enhanced our diagnostic capabilities in medicine. This field of liquid biopsy research is vastly expanding, with ctDNA or circulating tumor DNA research really leading this field. However, there are many other potential blood-based biomarkers, some of which you can see here, that can be found in the circulation and could be useful for a range of different clinical questions. And autoantibodies shown here in orange are one of those. Autoantibodies can be detected from either plasma or serum, although serum is generally preferred. Now, of course, they're better known for their involvement not just in the diagnosis, but also the monitoring of autoimmune diseases. But the link between cancer and autoimmunity has actually been established decades ago. And it is known that autoantibodies in cancer can have both an anti-tumor and pro-tumor effect, depending on which one you're studying. And from this point onwards in the presentation, I'm going to be referring back to this abbreviation whenever I talk about autoantibodies. Here's a nice timeline published by MacDonald et al., which highlights the main insights and discoveries that have shaped our understanding of this link between autoimmunity and cancer. As you'll note, the timeline only captures milestones achieved until the first introduction of novel immunotherapies, such as immune checkpoint inhibitors, that were first introduced in melanoma and lung cancer patients, but thereafter, the literature in this field expanded at such a rapid pace, it is actually quite difficult to summarize all the research findings that came after this point. Autoantibodies can arise from various factors such as chronic inflammation, infections, tissue damage, and even aging, but the precise mechanisms involved in autoantibody production in cancer are still not yet fully understood. We attempted to summarize the main causes of autoantibody production in cancer in this figure in the middle here. Since this was first published, a couple more theories have been produced, but personally I believe the causes of that antibody production will be different, not just between the different cancer types or even in individual patients, but also the antibodies that are studied themselves. Now, the general hypothesis is that tumors present or release tumor-associated antigens, which prime a humoral immune response, and antibodies are produced and then released into the circulation. It is interesting to note that these antibodies in cancer patients are often targeting intracellular proteins that perhaps have not been presented during the immunoediting process, and thereby they're elucidating an autoimmune response now toward this antigen that they come into contact with. Ultimately, autoantibodies may be suitable cancer biomarkers as they represent an amplified signature of the systemic immune response toward cancer. They're easily collected from blood, as I mentioned, and also measured, fairly straightforward, and they possess long half-lives compared to many other blood-based biomarkers. Here's a list of techniques that can be used for autoantibody detection. Now, this list is not extensive and it's not in any particular order of importance, but generally enzyme-linked immunosorbent assays or ELISAs remain the gold standard for antibody assays. So this would be an indirect ELISA format. But these generally only allow you to test one antibody at a time unless you're using a multiplexed immunoassay, which could be based on Luminex technology. That would be the most well known. So here you would immobilize your corresponding antigen on a bead rather than in a well on a plate like you would with an ELISA. And because you're immobilizing it onto a bead, there is the potential to measure multiple antibodies at once in the same sample. And depending on what access you've got to which instrument, in theory, you could measure up to a total of 100 or even 500 targets at the same time. Although I would argue that most research multiplex assays measure a whole lot less targets. Now, if you do have a study with a really well-defined cohort and would really like to run an in-depth omics scale discovery of your patient's serum to identify unique serum profiles of autoantibodies or shared autoantigens for your study cohort, then I would suggest the use of protein or antigen microarrays. Those microarrays have antigens immobilized on their array surface rather than on a bead or in a well on a plate. The patient's serum is then added, and any antibody that is bound to its corresponding antigen is detected with a fluorescently labeled secondary antibody that can detect different antibody isotypes. Most commonly, of course, human IgG is measured, but if you have access to a 2-laser microarray scanner, then you could also measure human IgM or A or any isotype you're interested in at the same time. And this essentially doubles the amount of data that you will get for each patient. There are many different suppliers and types of protein microarrays available, and they all have different lists of antigens for different reasons. But the main protein microarray that I have used and I'm still using and would therefore suggest for your discovery is called the human proteome microarray. Also known as the HuProt microarray, this array is available from CDI Labs, our host today. It is the largest protein microarray in the world and has been really well cited in the research literature. It covers over 81% of the human proteome, with each protein being printed in duplicate in their natural three-dimensional conformation on the slide. It also contains lots and lots of control spots, which is good for general data QC as well as data normalization. And as an assay, it takes around half a day to run. And I must be honest, most of this time is actually just waiting incubation time as well. Now, the protocol that is available online is really helpful as a starting point, and CDI Labs have been very supportive with all of our questions. Personally, I found the most difficult part is actually the very last step, the drying of the arrays just prior to scanning. But the team and I have worked out some tips and tricks that we're happy to share during question time if you like. I also like that there's a very detailed database available already on the web page of every single protein that exists on the array. So if there's a particular protein or perhaps a group of proteins you're interested in, you can look that up at any time to make sure it's represented on the array. And each protein is linked to its UniProt ID and many other databases. So this enables you to identify exactly which protein you may want to validate, and you can even order focused microarrays from CDR Labs for validation. Now those arrays allow you to measure antibodies in multiple patients on one array against a smaller selection of proteins of interest. So with a HuProt you would measure just the one sample per slide, versus with a focus array you would measure say 14 samples depending on which layout you pick against perhaps your top 100 or 200 markers of interest, which makes the validation a lot faster and also more cost effective. And it's really a nice next step in your research pipeline before you jump to other methods. I'll be discussing some studies using HuProts and other techniques and methods in the coming slides. Now, as I mentioned, there is a lot of autoantibody research available in various types of cancers, especially in breast, in lung, and in gastrointestinal cancers. However, for the rest of this presentation, I would like to focus on research into autoantibodies as biomarkers for cutaneous melanoma, or melanoma of the skin. Now, this is still a bit of a niche research area, and I'll try to summarize the majority of the research, but please be aware that this presentation may not cover every single paper and not all the literature in this field. For example, there are many published case studies that we just don't have time to discuss here today. So, unfortunately, New Zealand and Australia, which is where I'm located, are the melanoma hotspots of the world, but melanoma remains a major global health concern to date. It is often referred to as Australia's national cancer, which is not a great title to hold. And we know it's a potentially deadly type of skin cancer, but thankfully prevention and sun safety educational campaigns are making a difference. And the early detection followed by the prompt surgical excision of the primary tumor can lead to amazing 5-year survival rates, as you can see in the image. However, if the primary tumor is not detected, it may grow into deeper layers of the skin and metastasize to lymph nodes, which would make you a stage 3 patient, or the rest of the body, which is a stage 4. In the past, stage 4 melanoma patients used to be told that they only have about 6 to 9 months to live, with limited treatment options available, including chemotherapy, radiation, and also targeted therapies, which worked amazingly well at first, but then resistance kicked in and the cancer came back. But melanoma was one of the first solid cancers for which immune checkpoint inhibitor therapy was introduced in 2011. And now we have over a decade's worth of data available on both single agent and combination immune checkpoint inhibitor therapy data. And patient survival has increased to approximately 50% 5-year survival, which is amazing. In the following slides, I'll be discussing mostly published and some ongoing research on autoantibodies as biomarkers for cutaneous melanoma across the following areas of the disease spectrum. So let's commence with melanoma diagnosis. Here's a word map of the most commonly studied autoantibody targets for the diagnosis of 5 major cancer types, including melanoma. Now, I think it's not surprising to any of us that anti-p53 antibodies are in fact the most commonly studied and published potential diagnostic cancer biomarkers in this autoantibody field. This study by Lewis and colleagues in 1969 was one of the first to identify autoantibodies in the sera of melanoma patients and to observe that levels of these antibodies are higher in melanoma patients at early disease stages compared to more advanced disease stages. The authors found 2 types of autoantibodies. One type was active against antigens on the melanoma cell surface and another reactive with cytoplasmic antigens that are present in most melanoma cells. Now, the authors were actually unable to identify the specific antigens, but noticed matching sera and tumor reactivity in samples originating from the same individual. They also found that a melanoma-associated autoimmune response could be induced following the inoculation of 2 melanoma patients with their own irradiated tumor cells. Then in 2006, a paper by Nesterova et al. Proposed ECPK antibodies as potential cancer biomarkers. Now, this is an enzyme that is secreted into the circulation by many cancer cells, where it is then called ECPKA, or extracellular protein kinase A. This particular paper found that IgG anti-ECPKA autoantibody levels were found to be raised in a variety of 20 different cancer types, including melanoma. And this was interesting since 2 years earlier, Kita et al. In 2004 found that blood ECPKA levels actually decreased after the surgical removal of solid tumors. The sensitivity and specificity for detecting the incidence of those 20 different cancers relative to controls just using an ordinary ELISA for these antibodies was found to be superior actually to that of detecting the corresponding antigen, with the autoantibody assay yielding an area under the curve of 0.937 compared to an area under the curve of 0.869 for the antigen. However, as noted previously, these are not specific for melanoma, and therefore it is not suitable for routine testing of people at risk of melanoma. Then in 2009, a paper by Gnatic et al. Was published that I actually consider to be a key paper in the field, mostly for its very detailed description of the data analysis of protein microarrays and also the use of serum scores that I will explain a bit later, and quanti-normalization, although these days RLM or robust linear model normalization is preferred for protein microarray data. And we now have platforms like the POWER platform, which stands for Protein Array Web Explorer R, that are really useful for data normalization and general analysis, but of course you can use your own scripts in R as well. Now, in this particular paper, Gnatic et al. Screened a pool of 5 melanoma cancer patient sera known to react with cancer antigens by ELISA, and they compared various commercially available protein microarray platforms for seroreactivity with antigens such as MAGE-A4, P53, and SSX2, 3, and 4. Now, the study found, first of all, remarkable concordance, in fact 94%, between the ELISA and microarray data, and they showed that the pooled sera from cancer patients displayed a much higher reactivity with the antigens compared to the low reactivity of pooled healthy volunteer samples. Although it's not quite clear what melanoma disease stage was actually represented by the pooled serum sample. Now, the pooled melanoma sera antibody reaction was actually highest in this instance against SSX2. Then 1 year later, Shimbo et al. Reported to have detected BPAC1 autoantibodies via ELISA in both early and advanced stages cases of melanoma, and their levels were found to be significantly higher in the melanoma patients compared to the controls. In fact, detected elevated levels of BPAC autoantibodies were actually found in a third, or 33%, of their early stage melanoma patient cohort, although that was relatively small, and in 20% of their advanced melanoma patient sample, of which they had 37. However, 5 years later, and using the same ELISA assay as well as an increased and independent patient cohort, Gemblicher et al. Detected elevated levels of BPAC1 autoantibodies in only 2.3% of their patient cohort, or in other words, only 5 patients out of 179 that they tested actually tested positive for elevated levels of BPAC1 autoantibodies. The authors also found no significant difference in the autoantibody levels between melanoma patients and healthy control serum, and they also did not observe high levels in the early stage patients versus the late stage patients, so that unfortunately did not validate. Then in 2018, as part of my PhD, our team screened sera from a total of 104 early stage melanoma patients, as well as 105 healthy controls, against the immunome protein microarray, which contained 1,627 functional proteins. We identified a short list of about 130 antibodies of interest from these, and we derived at a combination of 10 of those diagnostic autoantibodies, which has since been published, and those also include p53 autoantibodies. The panel that we identified had a sensitivity of 79%, a specificity of 84%, and an area under the curve of 0.828 for primary melanoma detection. We called this the MELDX panel, which is now undergoing extensive validation, and we hope that it may serve as an additional tool to assist clinicians with the early detection of cutaneous melanoma in the future. Now, what was really exciting was the fact that we were able to detect elevated levels of these antibodies even at the earliest possible melanoma stage, which is the TNM stage 0 or in situ melanoma, where the tumor is located only in the epidermal layer of the skin and is less than 1 millimeter thick. In 2020, and using an immunoblood Uroline kit, Ini et al. Discovered the presence of high titers of antiganglioside antibodies that are shown on this slide to be present in predominantly primary melanoma. Now, I understand this slide might be a bit difficult to read, but basically every single graph is telling the same story. At the top, you've got your control group, the middle group is the primary melanoma patients, and then the bottom one is the metastatic melanoma patients. And essentially, in orange, we're detecting IgM and the antiganglioside antibodies, and in blue, it's the IgG isotype. So across the board, IgM antiganglioside antibodies were detectable in predominantly primary melanoma patients. Last year, there was an interesting conference presentation by an Australian researcher, Vice Alonso and colleagues. And at this meeting at the 2024 European Academy of Dermatology and Venerology Congress, they described that they have discovered circulating antibodies against especially cancer testis antigens via a microarray. And they identified IgG antibodies to 3 melanoma-specific antigens with great accuracy overall, but an external cohort actually showed that another signature of 7 antigens yielded results of 83% sensitivity and 84% specificity for early stage melanoma detection. Next, we're going to look into autoantibodies as biomarkers for melanoma prognosis. In 2022, our team published a review article which summarizes the autoantibodies that have been suggested to be prognostic biomarkers for the 6 major cancer types, including melanoma. Now, a red triangle next to a listed antibody indicates that this biomarker has been associated with poorer prognosis in the described cancer, and a green triangle indicates a better prognosis. And in the coming slides, I will go through some of those examples. In 2011, Sabo et al. Used a protein microarray and demonstrated that stage 3 melanoma patients who were positive for autoantibodies against LDH beta, CT, SD, and SA AH1, these three glycoproteins, were significantly more likely not associated with nodal disease. So we're looking at healthy volunteers and non-melanoma skin cancers as a group. Here we've got melanoma patients with a negative node status, and the same again with a positive node status, as in the cancer was found in the lymph nodes. Now, of the 32 patients who were sentinel lymph node positive, 9 actually had elevated titers of a fourth autoantibody called GRP94. And of those, 9 patients demonstrated regional metastasis, while a further 4 of them demonstrated microscopic disease, even in nodes that were thought to be clinically negative. So the final established model, which suggested the use of all 4 of those autoantibodies in combination, with 3 of them suggesting a negative node status while 1 is a positive node status, achieved an area under the curve of 0.869, and it's thought to be useful in trying to find out who's likely going to develop regional metastases. Using ELISAs, Kerner et al. Screened a large cohort of 230 melanoma patients undergoing interferon therapy for the occurrence of CD28 autoantibodies, and they found that the presence of those elevated anti-CD28 antibodies actually correlated with a decreased overall survival. In addition to measuring this large cohort, they also had 6 patients with longitudinal follow-up samples, and 1 female shown here actually had 10 years worth of follow-up. Now, as you can see, she was initially diagnosed with primary melanoma in 1998, and then over time developed metastases, first to lymph nodes, then the lung, and then brain and liver. And she had progressed disease status scores from her scans before the final point occurred. Now, at this point over here, where she gets her lung metastasis, this is the point where we would then call her a stage 4 melanoma patient, and that seemed to coincide with a steep rise of anti-CD28 autoantibodies, which was interesting. According to May and colleagues, the development of autoantibodies as part of spontaneous autoimmunity as a consequence of a break in self-tolerance is actually an independent good prognostic factor for stage 4 melanoma patients. In this study, a total of 103 patients were studied, with antithyroid and antinuclear autoantibody assays being performed every 6 months. Now, any detectable occurrence of spontaneous self-antibodies at the upper detection limit of those assays, at least for one of those assays, was considered to be a biological marker of autoimmunity. Now, the authors found that patients whose immune system had generated spontaneous self-antibodies actually had an increased median survival for 40.7 months instead of just 30.4 months. 2 years later, Zaenker et al. Also researched the prognostic significance of spontaneous antibody responses against tumor-associated antigens in melanoma. They utilized a Luminex speed-based assay to investigate antibody responses against 29 tumor-associated antigens in a total of 365 melanoma patient sera, including all disease stages, as well as 100 gender-matched healthy controls. Now, out of the 29 targets tested, anti-recoverin autoantibody responses were found most frequently in melanoma sera of all disease stages, and antibody responses against specific tumor-associated antigens in stages 1 to 3 melanoma patients correlated with poor prognosis and or a shorter progression-free survival, while in stage 4 patients no such specific association was identified. In the diagnostic section in this talk a little earlier, I mentioned the MELDX study that comprised of 104 primary melanoma patients, all the way from in situ stage 0 to including TNM stage 2 patients. Now, we were able to actually follow the outcomes of 81 of those patients over a period of about 6 years. And although they were diagnosed with early stage melanoma at the time of when we first collected the blood, 10 patients actually had progressed with their disease and 71 did not progress. So we looked at our original immunomicroarray data, this time separating our groups by disease progression status with the hope to identify some autoantibodies that might have been predictive of this cancer progression in the future. And we then went ahead and developed a bead-based assay for the marker of interest for future validation. Now, when looking at all 1,627 targets, there was no difference in the number of positive antibodies between the two groups. However, when we're looking at a short list of our identified markers, then we can clearly see that the number of antibodies was a lot higher in the progressed group versus those who did not progress, and that remains the same for the serum score. We've also identified two markers of interest, one of which we decided to do further research with. Now, you've heard me mention that serum score that I just talked about when I described the Genetic paper from 2009. So this is a nice score that can be used to describe seroreactivity for multiple antibodies in one sample. And it can be useful when comparing seroreactivity from one sample to the next. So the serum score is the sum of all the RFU or relative fluorescent units readings of the particular antibody that made it above their specific cutoffs. So we coated our own beads and developed a bead-based assay for the detection of those marker 1 autoantibodies. Now, we could have developed our own in-house ELISA since we were only interested in one marker at a time, but we chose to develop bead-based assay with the idea of potentially adding more markers in the future. Unfortunately, there was one sample we could no longer find, so it's down to 80 samples for this new assay. But in general, you've got your microarray data on the left-hand side and your bead-based assay results on the right-hand side, and as you can see, the results are very comparable. Even their ROC curves look almost identical. Now, that was exciting. However, when we do compare the classification call for each individual patient as either belonging to the progressed or non-progressed group, for each detection method, we only achieve around 60 to 70% concordance. And we are currently looking further into this, and I'm happy to discuss this a bit further during question time, perhaps. Next, I'm going to discuss the need for and the evidence of autoantibodies as melanoma treatment response prediction biomarkers. As I mentioned before, the 5-year survival response rate of metastatic or stage 4 melanoma patients has drastically improved, and many patients now live many years due to the introduction of immune checkpoint inhibitors or ICIs, such as ipilimumab, which is an anti-CTLA-4 inhibitor. Then we've got pembrolizumab and nivolumab, anti-PD-1 inhibitors, and also the combination of ipilimumab and nivolumab together. Overall, the function of these drugs is to block immune checkpoints that are used by cancer cells like melanoma to evade an immune response, and this will enable an active immune response against melanoma tumors, and some amazing responses have been observed over the years. However, despite this, there's still around 40 to 50% of all patients that just still don't respond to these treatments, and it is very difficult to predict this from baseline. Therefore, the search for suitable biomarkers, including blood-based or serological biomarkers like autoantibodies, continues. Here's one study by Fessler et al. From 2019. In the study, 2 independent patient cohorts with a total of 41 patients undergoing ICI treatment were investigated. Now, the patients were classified as either responder or non-responder as per RECIST guidelines. Analyses were used to measure a specific IgG antibodies against 5 different melanocyte differentiation antigens, of which I'm showing one here as an example. They found that higher baseline antibody levels were significantly associated with better clinical response to checkpoint inhibitor therapy. These antibodies also correlated with a longer overall survival and progression-free survival in both cohorts. Now, interestingly, the antibody level remained stable or even increased in other examples in the responders, while they either remained stable or decreased in the non-responders. And the authors thought this could be used as a complementary biomarker in addition to PD-L1 status, for example, tumor infiltrating lymphocytes and or tumor mutational burden as well. This was also one of the first studies that were looking into antibody levels early during ICI treatment as well, not just baseline. Then last year, I was very excited when I read this paper in Nature Reviews Immunology that discussed the potential biomarkers for combination immunotherapy. Now, in the blood biomarker section, it was actually proposed that autoantibodies may be potential future biomarkers for combination immunotherapy. Our team also screened a cohort of 100 metastatic melanoma patient samples at baseline for ICI treatment response, and we screened these samples for both IgG and IgA autoantibodies using the HuProt microarray, which, as you may remember, enabled us to investigate over 21,000 potential targets for each patient. Now, this is not yet published, but so far, after performing data QC, normalization, and differential expression analysis, and then devising a short list of markers of interest, we found that there was not much overlap between the antibodies identified in each treatment subcohort. But responders do certainly have higher levels of those baseline autoantibodies than patients who did not respond to the treatment. Now, the identified antibodies were involved in a range of biological pathways, some of which are vaguely listed here, and they were generally associated with cancer or immune responses. We also found that patients with an elevated serum score for multiple antibodies at baseline had a significantly better overall and progression-free survival than those who had a lower serum score. Since then, I was able to acquire some more funding to enable us to screen another cohort of 96 metastatic melanoma patient samples, again at baseline and the same treatment. And this was done in association with researchers at Macquarie University and the Melanoma Institute Australia. We used HuProt microarrays again, and we're currently analyzing this data set. Now, having used HuProts for this omics scale discovery of autoantibody profiles in 2 independent cohorts, the next step is then to order focus arrays that will allow us to measure even more samples, which we already have, as well as additional time points other than baseline samples to get a better idea of the identified autoantibody dynamics over the disease course. Ultimately, we seek to use this validation to define an even smaller panel of autoantibodies that we can then develop into a multiplex bead-based assay. Since Luminex platforms are already located in most routine pathology labs, and therefore TGA, which is the Australian version of FDA approval for our assay, may be easier to get in the future. We envision that this may be one suitable pathway to bring this new cancer autoantibody assay to the clinic one day. Finally, I would like to summarize what has already been published in this relatively new field of autoantibodies as predictors of ICI toxicity in melanoma. A couple of years ago, our team published another review paper that summarized studies on autoantibodies as toxicity biomarkers in a range of cancers. Now, I understand that this slide is very busy and difficult to read, but here I have essentially listed all the melanoma studies from this review paper, and I will touch on some of these in the final slides. Essentially, immune checkpoint inhibitor therapy has been a game changer for advanced stage melanoma, and it is now even used in the adjuvant treatment setting. But the occurrence of immune-related adverse events is common, and especially when using combination immunotherapy. The side effects are often autoimmune nature, and they're graded from 1 to 5. The occurrence of mild side effects throughout treatment have been associated with better treatment responses and even survival, but grades 3 and 4 are indicating severe side effects that often cause the cessation of potentially life-saving treatment from an oncology standpoint, and some are even irreversible. In general, severe immune-related adverse events will require hospitalization and the prescription of high-dose corticosteroids that will actually reverse the effects of what ICI treatment tried to do. And grade 5 are fatal events that are rare but can occur. So as you can see, there's an urgent need to be able to predict the likelihood of those side effects in patients about to undergo ICI treatment. And it is believed that some patients actually harbor pre-existing antibodies or subclinical autoimmunity that might even predispose them to the development of those side effects upon immune checkpoint inhibition. And therefore, autoantibodies are certainly of interest. For example, using the immunome protein microarray, elevated levels of autoantibodies in the blood of 5 stage 3 and 4 melanoma patients who received CTLA-4 immunotherapy combined with an experimental intralesional injection showed value in predicting the occurrence of side effects prior to their treatment commencement. So before the side effects were actually clinically apparent throughout the treatment in 2 patients, which are shown in red, compared to those who did not have side effects shown in black. Unfortunately, this trial was discontinued after the recruitment of only 5 patients though. In 2018 again, and using the HuProt microarray this time, Gowan et al. Identified baseline autoantibody profiles in melanoma patients on CTLA-4 or PD-1 immunotherapy, as well as some on combination immunotherapy. And those were predictive of severe immune-related adverse events toxicity. Now, the list of their top 15 differentially expressed antibodies for each treatment group are shown here. And pathway analysis revealed that significant enrichment of antibody targets associated with immunity or autoimmunity, including the TNF-alpha signaling, toll-like receptor signaling, and even microRNA biogenesis were identified. This is again a key paper in the field. However, the study only tested 11 patients undergoing combination immunotherapy, which was representative of the time when the paper was published, but is now used a lot more commonly, and the focus was on baseline samples only. Then in 2020, there were 2 ASCO conference abstracts from 2 groups that discussed toxicity prediction using autoantibodies. The first abstract by Hassel and colleagues presented data at the ASCO meeting that showed that baseline autoantibody profiles in 333 patients with metastatic melanoma on ICI treatment using a custom protein microarray of 832 cancer and immunity-related antigens were predictive of treatment response. They also identified anti-MAGE-B4 autoantibodies to be associated with higher immune-related adverse events incidence, and anti-FGFR1 antibodies to be associated with fewer side effects. Then, using patient samples from the CheckMate 238 phase 3 trial, Osman et al. Identified autoantibody signatures that could accurately predict toxicity requiring treatment discontinuation. The model showed a strong predictive performance. They've identified a signature of 170 antibodies, 102 for the ipilimumab arm and 68 for the nivolumab arm. And it was interesting to see that many of these corresponding antigens were actually nuclear protein targets. A more recent paper by Johanna et al. Also utilized the HuProt microarray in an impressive total of 950 patients from 2 clinical trials. Now, they identified baseline autoantibody candidate lists that were then converted into a risk score for the prediction of melanoma recurrence and side effect occurrence in the adjuvant immunotherapy melanoma treatment setting. Now, the identified list of autoantibodies contained hundreds of entries, which again shows that autoantibodies generally need to be assessed as combinations or panels instead of as individual biomarkers. But those lists do require further validation, and they may be difficult to translate into the clinic in their current form. Finally, we also looked into ICI toxicity and divided our original cohort of 100 metastatic melanoma patient samples into 2 groups, those with severe side effects and those with either no or mild side effects. We found a profound IgA signature in the group with severe side effects and identified multiple IgA and IgG antibodies from our HuProt data set for further validation using focus arrays. So in conclusion, we know that this field of autoantibodies as cancer biomarkers is rapidly evolving. Autoantibodies are a biomarker that is easily collected and tested, and they reflect an amplified systemic immune response to cancer. Autoantibodies need to be tested in combinations, and the methods used for discovery are really powerful. They may be a little expensive and perhaps difficult to analyze, but it's certainly worth it. For validation, I suggested the use of focus arrays, and we do need to be able to convert our findings into immunoassays other than protein arrays for routine testing later on. Now, the optimization of those assays could be quite difficult and take some time, however. We generally need to understand the autoantibody dynamics over time a little bit better. And so far, there are no official guidelines on how to best analyze and also report protein microarray data. There's lots of publications and advice, but it would be great to have an official guideline available. There's a real opportunity here, though, for translation. I mean, immunoassays are already part of routine testing, and our research could give us many other interesting insights into the disease that we're studying as well. With that, I would like to thank all study participants of the mentioned research studies. I would also like to thank the cancer autoantibody research community in general, as well as our research team pictured here and my students in the image down the bottom. You are all an integral part of the Centre for Precision Health at Edith Cowan University in Perth, Australia. Finally, I would like to thank all collaborators and funders of our concluded and ongoing research projects, and thank you especially to CDI Labs for hosting this webcast and for Springer Nature for providing this platform. And of course, thank you to you for your attention. Please feel free to contact me via my email address at any time after this webcast. Thank you.
Thank you so much for your presentation, Dr. Zaenker. It's now time for the Q&A. If you'd like to ask a question, just type it in where it says questions and then press submit. All right, I see we've already got a few questions in here. So someone was curious, what other research is currently being conducted in the space of melanoma and autoantibodies?
Okay, yeah, thank you very much for that question. Obviously, my presentation was, had time constraints. So as I mentioned, I couldn't mention everything, especially what we do in our group. But with that, I really need to acknowledge Professor Mel Zimmern, who was my principal supervisor in my PhD, who even got me interested in this field as well. So in terms of what we do, we have many other studies on on the way at the moment. For example, we are analyzing, again, the HuProt data set that I first mentioned of our initial 100 melanoma patients for the detection of, or the potential to detect brain metastases from blood. So this is a PhD project of my student Desiree Sexauer, who is using a multi-omic approach. So she's looking at the autoantibodies as well as methylated DNA and also microRNA as well. And we found so far that IgA isotype antibodies don't really seem to be useful for this question, but certainly IgG, and she's already discovered a short list of those. And I'd just like to mention one other study out of the many we're doing. So when I first described the 100 metastatic melanoma patients that we initially screened on the HuProt array, I only spoke about baseline samples. But out of the 47 patients who received pembrolizumab treatment, 25 of those had a baseline sample as well as a second time point screened against the HuProt. Now that time point was at 6 to 9 weeks post commencement of the treatment. So we call that an early during treatment time point. And what my other student, Rebecca, is currently doing for her master's is she's looking at the start of only those 25 people, and she's calculated the delta fold change between the baseline and the early during treatment levels. And she's deliberately looking for antibodies that increase their level early during treatment in responders. And she's already got a list of markers of interest. And her top marker is an IgA isotype antibody that has a sensitivity and specificity of over 80%, which is quite amazing as a single antibody. So we're currently developing a bead-based assay just for that one, and we will add others as well.
Great, thank you. Someone else was curious, will it be difficult to translate any of the mentioned findings into the clinic? What steps are required?
Thank you. So that is the most important question, because ultimately I hope we all end up with a beautiful assay that actually makes a difference in the world for sure. Now, interestingly, there's actually a really nice paper in the autoantibody autoimmunity field, not cancer, but just general autoantibody assays, from which we always learn anyway. So this paper was by Fritzler et al. From 2021, and they basically summarized all the steps, but very briefly, what they're essentially saying is once your research and development, your initial phase is concluded and you're clear that you've got a nice discovery, really important to protect your initial discovery with, for example, getting a patent. And some groups also start to do tech transfer discussions with particular companies, or maybe commence their own startup if that's an option to them. Then, of course, you need to keep developing your assay and ultimately validate what you found. And ideally, you're going to end up with nice clinical trials where you're comparing what happens if you're adding your blood test on top of what is gold standard. And then you need to get regulatory approval. And once you get that, which is easier said than done, it will be out in the world. But there you are also, there's many other challenges, because then your assay needs to basically compete with everything else that's out there. So there's many different hurdles, but I like this paper that I just mentioned, because they give you a particular criteria that lets you assess how good your potential, like how viable your assay might be. It's called the SMART criteria, and it's spelled a bit funny. It's got 3 S's, 2 A's, and 2 T's, so it looks strange, but it's very clever. So it essentially says you think about your assay and you got to assess whether it is obviously sensitive and specific. We know that. Also think about, is it scalable? And is it measurable using conventional techniques that are already out there in the pathology labs? Then you need to think about whether your test is actionable. Will it actually change anything in the treatment? And also, will it add value? And then finally, you need to think about whether or not it's realistic, what you're trying to measure, or is this a really difficult thing you're trying to measure and only you alone are the one who can analyze this data, or can other people do a similar thing? And then finally, you got to think about your temporal timings of your antibodies, because in the autoimmunity field in general, it is known that autoantibodies are not always present at baseline. So when you first diagnose a disease, and sometimes they persist during treatment and sometimes they don't. So once again, we're really having to understand what happens to these levels over time. And if I may, I just want to mention one really good successful example of an assay that has achieved all of this. So I guess the most famous cancer-associated autoantibody test out there right now is called the early CDT lung test. As the name suggests, it is a lung cancer diagnostic, and it's supposed to help predict risk of lung cancer. It's a semi-automated ELISA, and it measures 7 autoantibodies. And once again, P53 is also in that panel. And basically, it is supposed to be used in conjunction or in addition to CT screening, which is the gold standard for lung cancer detection. And they've already done one really huge clinical trial of over 12,000 people in Scotland. And over a 2-year follow-up period, they actually found that the people in the arm of the trial that have received both the blood tests and the CT scans actually showed a 37% reduction in their late-stage presentation of lung cancer, which is amazing compared to those who only had the scans. So here's just a really good example that it can be done.
How important is it to identify autoantibody types and subtypes?
Pardon, can you repeat that, please?
Sure. As someone was curious, how important is it to identify autoantibody types and subtypes?
Good question. So I would argue quite important. For example, just talking about the isotypes again, generally we tend to just measure IgG, which is really important, obviously, for many different immune reasons. But I actually found many examples of where we actually found IgA to be more predictive of certain clinical questions than the IgG. And personally, I actually believe that ultimately there might be a perfect panel one day, or near perfect, let's call it that, that doesn't just consist of one type or one isotype. I believe ultimately there might be a particular panel that measures all sorts of different types, and yeah, that might be optimal ultimately, because that reflects what happens in the human body.
How do autoantibodies perform as biomarkers compared to circulating tumor DNA?
Yes, so our team is actually, we have a long history of being involved in circulating tumor DNA research, not me personally, but all of my colleagues. So ctDNA is a brilliant marker for monitoring of cancers over time in particular. And as I mentioned in my first few slides, it is absolutely the leader in the field. It is the most advanced liquid biopsy biomarker without a question. Now, we actually don't so much compare them. We actually want to use them together. So with that one study I mentioned where we're looking at baseline versus early during treatment time points, even though it's only a small cohort, I actually forgot to mention that on top of looking at those autoantibody changes, we're actually overlaying ctDNA results as well. The idea is as you're starting to respond to treatment, your ctDNA levels will drop and perhaps the antibody will rise, and that means you'll respond. That's just one hypothesis. But I think they all have different biological meanings. Of course, with ctDNA, you can measure specific mutations, versus with your antibodies, it's a bit more of a systemic response you're measuring, but they are also target specific. So I just think they tell us different things.
You have longitudinal samples of patients receiving immunotherapy to evaluate the induction of de novo autoantibodies. If so, are they associated with good or bad clinical outcome?
Thank you. Brilliant question. So the answer is yes, absolutely, we do. So our team is actually, we're biobanking melanoma samples, and we've been collecting melanoma blood samples at all sorts of disease stages and treatments, and in particular immunotherapy over years. So yes, we do. Haven't looked into it just yet. So far, we've only compared, again, those baseline and early during treatment samples, which is one other time point, simply because, as I mentioned, the discovery, which is usually done by, say, a HuProt, for example, is crucial, but it can be a little bit expensive. So you don't really, that's not the right time to measure many different time points from a particular person, I would argue. But then once you get to the immunoassay stage, I would say definitely you've got to look at what happens longitudinally. So yes, that will definitely happen, but we're not quite there just yet.
We have time for one more. Given that antibody responses, for instance, B cell responses, require CD4 plus T cell help, and CD4 cells also help prime CD8 T cell responses, I wonder if TAA specific autoantibodies can serve as markers for CD4 and CD8 T cell responses in cancers.
Potentially. So I'm not, I can't off the top of my head think of a specific study that has necessarily compared just that. But yeah, I guess it could be an indirect biomarker. I agree with that. And also should be mentioned that many other groups are, on top of looking into autoantibodies, also measure B cells and B cell subtypes as well, because ultimately those turn into plasma cells that then actually release the antibodies. So yes, absolutely, there's a lot more cell-based work to be done as well. And then what I keep getting as feedback for my grant submissions is it would be great to do functional studies on your antibodies, and I totally agree, but we need to get to a particular marker of interest that we really want to study in detail. So I'm hoping that in the next few years we'll get there.
Well, thank you so much for your time and for your presentation and for answering everyone's questions today. And of course, a big thank you to our audience for being here with us. I'd like to remind everyone that you can watch this webcast again at any time on demand at nature.com/natportwebcasts. And thanks for watching, and I hope you'll join us soon.
Thank you.